All right, hello everybody. We're going to talk about DNA replication tonight. To replicate something means to copy it. So we're talking about DNA copying itself before the cell can divide. If you remember our cell cycle, that happens during the S phase of interphase. So DNA replication happens during the S phase, specifically so that a cell can divide and so that there's identical copies of DNA in the two new cells. So let's talk about how that happens. So first of all, before we get there, let's talk about another historical experiment. Remember that scientists, even today, scientists can barely see a DNA molecule using microscopy, electron microscopes. But back in when this happened in the 1950s, they couldn't see anything more than the chromosomes. They could not actually see how the DNA was replicating itself. So the scientists were trying to hypothesize, well, what are some models? What are some things that could explain how DNA would be copied? And then they Meselson and Stahl came up with an absolutely amazingly beautiful, ingenious experiment to test these three models. So there's three possible ways that DNA was replicating itself. The first idea was called semi-conservative, where each original strand, we knew that we had kind of opposite strands, where each original strand was separated and then each strand was copied. So take a look at the diagram here. The red shows the original DNA, and then the pink is the new DNA. So you have two DNA strands that are a hybrid or a combination of the new and old from the old strand. So that was one possibility. So the result is each DNA molecule has one original and one new strand. The next model or possible hypothesis was conservative. Both sides were copied and the molecule was left intact. So you end up with one completely old DNA molecule, that's in red, and one completely new DNA molecule, and that's the one in pink. The last hypothesis was called dispersive, and that's where sections of the original DNA molecule were left intact and then sections of new DNA were attached to it. So each molecule had sections that were original and sections that were old, and you can see that in the diagram here. So Meselson and Stahl come up with this absolutely ingenious way to figure out which of those is correct. They can't see the DNA. So this is what they do. So they grow some bacteria with an isotope that's called heavy nitrogen, and it's nitrogen 15. All of the bacteria that are grown initially have been grown in this nitrogen 15. So that means that all of their DNA, which has nitrogen in it, has this heavy isotope. Some of the bacteria then from this, what's growing, this sample here, are then placed in a centrifuge. They're blended all up and spun around, and they discover that the DNA is located as a band in the test tube itself. So take a look at this diagram here. So the DNA is right here. That's where the DNA landed, and it's got this heavy isotope, a heavy nitrogen isotope in it. So then that was before there was any chance for the DNA to replicate itself or anything. So we call this generation 0. So then the scientists remove them from their nitrogen 15 medium and grow them immediately in a lighter version of nitrogen called nitrogen 14. So now, as the bacteria replicate their DNA, as they copy their DNA, any new DNA is going to have a lighter mass. It's going to have less mass because it's going to contain the lighter nitrogen instead of the heavier nitrogen. So again, they take a sample, they centrifuge it, they spin it around, and then they look for where the DNA is this time. So this time with generation 1, so the first, it's kind of confusing because you have generation 0. Those are the ones that grew in the completely heavy nitrogen. Generation 1 is the ones that have been transferred to, they're the first generation in the light nitrogen. So it says generation 1 shows a single band again, but it's somewhat lighter than the generation 0 band. So which hypothesis right away by generation 1 can already be ruled out? And the answer is the conservative model. And if you can't remember what the conservative model is, it was the idea that you would have one old DNA and then one completely new DNA molecule. So both strands were old and both strands were new, because that would produce two banding patterns in the test tube. You would have one group of DNA that were light, and you would have one group or one band of DNA that was full of the heavy nitrogen, and one band of DNA where the DNA was made of the light nitrogen. So immediately by generation one, we know it's not conservative. We can't see the DNA. We're just figuring out whether the DNA is heavy or light. So then the bacteria were allowed to replicate several more times. And you can see what the banding pattern was in each test tube after each replication. It shows you the minutes down below. Bacteria replicate roughly every 20 minutes. So by generation 2, the second generation growing in the N14, we can already rule out one of the other hypotheses. So which hypothesis can be ruled out by generation 2? The dispersive. If you look at the banding pattern for generation 2, it shows that one is a mid-level mass, like it's a combination of nitrogen, heavy and light nitrogen. But then there's a band that is made entirely of light nitrogen. Well, that doesn't work with dispersive. Dispersive was the one that would have some chunks of completely old DNA attached to some chunks of completely new DNA. And if that were the case, you would never have a completely light band of DNA. It would always include, in the dispersive model, it would always include some DNA that had this heavy nitrogen. So already we've concluded that dispersive and conservative aren't possible. So as the generations continued, it became clear that semi-conservative was the correct model for the DNA replication. Here's another image kind of explaining what they saw. So in generation 0, look right under my photo here. I'm trying to point to it. It's so awkward to point on this because my image is reversed for some reason. So right there. Okay, so that's the original bacteria that were grown in the heavy nitrogen. That's their DNA banding pattern, and it's heavy. So then they go through one round of replication, and now we know what's going on. They didn't know at that time. So what happened is the two heavy bands separated, and each one was copied with light nitrogen. So look at by generation 1, we should have two medium mass DNA molecules. What that means is half the strand is heavy and half the strand is light, and the other one, half the strand is heavy and half the strand is light. So they would have a medium mass. So that's sure enough where that banding pattern is with generation 1. We have right in the middle a medium mass. Then by generation 2, we still have the medium mass. Those guys are right there. They're right there. But now they've split. And so now we have two DNA molecules that are made entirely of light nitrogen. So they're going to form the really light band at the top that has no heavy nitrogen in it at all. But we're still going to have those two right here, this one and this one, that have half heavy nitrogen. And so if you look at the banding pattern, here's the light nitrogen pattern, and here's the medium nitrogen pattern, the ones that have a light and a heavy banding pattern. And then as you continue through the generations, more and more of the generations are light, and there's always this small amount that have that heavy original DNA. All right, so that's a super cool, ingenious experiment that they did in the 1950s. Now let's move on and show you what we have learned about how DNA replicates itself. So this is a super cool image. It's actually of a DNA molecule that is replicating. And we're going to use three terms to talk about sort of the three stages of DNA replication. The first part is initiation. So that's how DNA replication gets started. Then we'll talk about elongation, which means elongating the copy. And then the last one is termination, how we end it, how DNA replication comes to an end. Okay, so initiation is what we're starting with. So it says replication can only begin at specified or specific locations. They're called origins on a chromosome. Once replication begins, it proceeds in two directions from the origin. So take a look at this squiggly noodle of a line. That's the DNA. And notice there's like a bubble, and that's because the DNA is being copied and it's moving in two different directions. This image will make more sense in just a second. So it goes in two directions at the same time. So here's what you were actually looking at, and you can see this image here and here, but let me talk you through it. So we've got prokaryotes on the left, and we've got eukaryotes on the right. The really important difference between prokaryotic DNA and eukaryotic DNA, let me say the similarity first. They both use A, T, C, and G. They both have nucleotides. They have sugar phosphate backbone. The DNA is almost identical. The difference is that bacterial DNA is a circular chromosome and eukaryotic DNA is a linear chromosome. And that becomes important as we're trying to do replication. So let's look at the left side. So there's an origin of replication. I'll try to point to it with my arrow here. There's an origin of replication. And once it opens up and the DNA starts getting copied, the DNA copying proceeds in two directions. So look at the arrow. It's going two different ways. And the replication is the part that's being copied, the part that is new is in light blue. So you see the light blue is increasing. There's more and more light blue, and then eventually they separate from one another. And it's two circular molecules that are made. You can see it happening right here. This is the DNA molecule, and the pink arrows show what direction it's going. It's going around that way. The linear eukaryotic DNA is a little bit more confusing. So with the linear, there are many origins of replication, which means it starts simultaneously in several locations. So it forms these little circles that we see. Those are called replication bubbles. And so there was an origin right here where there's a dot, and there's an origin right here where there's a dot, and there's an origin right here where there's a dot. And so the DNA separates. The light blue indicates where it's being copied, and it's moving this direction. Eventually the bubbles bump into each other, and that's when it kind of stops for that particular section. So it says there's some important enzymes that you need to know that have to do with replication initiation. The first one that I'm mentioning is arguably the most important of the enzymes in replication, and it's not in this diagram yet. So DNA polymerase, there are, I think in eukaryotic cells, there's like 15 DNA polymerases. That's an approximate, different types of DNA polymerases. And I think in the prokaryotes, there's like 5 different types of DNA polymerases, and I'm just going to call them DNA polymerase. Some of them have Roman numerals by them. We're just going to say DNA polymerase for right now. Okay, DNA polymerase's job is to actually attach one at a time the opposite nucleotides. So if the original strand of DNA says A, then it's going to attach a T. And if the next one says C, then it's going to attach a G. That's what DNA polymerase does. Helicase's job is to unzip the two. Remember that they are the A's and the T's of the original parental strand of DNA are bonded with very weak hydrogen bonds. So helicase comes through and it separates the A's and T's that are opposite from each other. And then topoisomerase, which is up here, remember that DNA is a double helix. It's all twisted up. So topoisomerase is kind of untwisting it. And then helicase, I said that wrong. Helicase is unzipping the two sides so that now these other enzymes can come through and start copying one side or the other of the DNA. Now, I mentioned primase because it's in the diagram, but the College Board has specified exactly which enzymes you need to know. You need to know DNA polymerase, helicase, topoisomerase, and then another one that's coming up called ligase. You do not need to know primase, but it shows up in a video we're going to watch, and you might stumble across the words. So primase hops on and it attaches what's called an RNA primer. And basically, you can see it in red. It's like a little signal to DNA polymerase, hey, this is where you begin. DNA polymerase, come on over, start right here. So without the primer, DNA polymerase won't click on. So that's primase job. It's priming it to get it ready for DNA polymerase to come through. All right, so now we're going to get, that was initiation, that's starting of DNA replication. Now we're going to actually start elongating the molecule. So it says DNA polymerase, the really important one, adds nucleotides to the 3' end of the new strand. Okay, KJ, what does 3' mean? Well, give me a second. Let's take a look at this diagram. I like this particular diagram right here because it shows you that the two strands are anti-parallel, that they're opposite of each other. They all show that, but I think this one did a good job because they wrote the nitrogen bases upside down, just to give you a sense of, okay, yeah, these are upside down from each other. So notice this end is called the 5' end, and on the other end, it's the 3' end. But then notice on the other DNA backbone, it's exactly reversed. So the two strands are running in opposite directions. Now, where does the name 5' and 3' come from? The 5' and 3' refer to the numbered carbons on the deoxyribose sugar. So this is the deoxyribose sugar. Remember, it goes phosphate, sugar, phosphate, sugar, phosphate, sugar, all the way down the backbone. So here's a close-up of a phosphate and the sugar, the deoxyribose sugar. The carbons are numbered. So here's carbon number 1, carbon number 2. Remember, this goes back to your biochemistry unit. They don't necessarily show you the carbon at an intersection where two lines come together, where there's a vertex. There's a carbon there unless it's specified that it's not a carbon. So there's an O here, and that's how we know it's not a carbon. But any other time that there's two coming together, that's a carbon. So this is carbon number 1, 2, 3, 4, and 5 is above the ring, and that's what the phosphate group is attached to. So that's where we get the name 5'. So look at how the phosphate is attached to the 5th carbon on this. Now look at the other end. Down here, the hydroxide group is attached to the 3rd carbon, and that's because it's on the opposite end. So the hydroxide group gets attached to this is the hydroxide group, and it gets attached to the 3rd carbon. Now, how much of that do you really need to know? Really not any of it, but students always want to know, what does 5' and 3' refer to? It refers to where the phosphate is attached, and it refers to where the hydroxide is attached, and that's where those names 5' and 3' come to. Okay, now let's back up. The first sentence here said, again, DNA polymerase adds nucleotides to the 3' end of the new strand of DNA. So we're going to assume that this left side is the new strand. So DNA polymerase is always adding it down here to the 3'. So if there's another letter, it's going to add it underneath this thymine. It's going to add the next letter. Let's look at the next slide because it shows it a little bit better. So the other thing that I'm just reminding you of, it says, also remember what a nucleotide is. We haven't had this since biochemistry. So a nucleotide includes a nitrogen base. I think students really easily, I know I did, get the word nucleotide and nitrogen base mixed up. So a nitrogen base is just the molecule A, T, C, G, or U in RNA. We're not going to see any U's right now. And they're the letters that are opposite each other. So you can see the A, T, C, G, whatever. Okay, so a nucleotide is one of the nitrogen bases, A, T, C, G, or U, a phosphate group, and a sugar. And if it's DNA, it's going to be deoxyribose. And if it's RNA, the sugar is going to be ribose. So right now, we're only talking about DNA, so the sugar is always deoxyribose. So I would show you, this would be a nucleotide right here, the phosphate, the sugar, and the letter A. Here's another nucleotide, phosphate, sugar, and the nitrogen base C. Phosphate, sugar, and the nitrogen base G. Here it is again. This is the phosphate. 2 of the phosphates get lost. So phosphate, sugar, and the nitrogen base thymine. Okay, so then it shows you DNA polymerase removes those 2 phosphates and then attaches this nucleotide to the 3' end of the new side. Do you see that got attached to the 3' end? So now there's a cytosine down here that doesn't have a partner. So now DNA polymerase is going to add a guanine right here to the 3' end of the new side. So we say that DNA replication occurs in the 5' to 3' direction. 5 is at the top of the new strand and 3 is at the bottom of the new strand. All right, so here's a huge problem. And when you first learn about this, it's super confusing. Since the 2 sides of DNA are anti-parallel, I like to draw the analogy of them being one-way roads that are pointing in opposite directions. DNA polymerase is only allowed to go, it builds from 5' to 3'. That means that it's reading the old DNA 3' to 5'. Let's make sure that makes sense. So let's take a look at this other picture from the previous page. So yes, it's adding it's adding to the 3', but that's the 5' end of the other molecule. Where it started was the 5' of the new molecule, but it started at the 3' end of the old molecule. So it's moving from the 3' to the 5' of the old molecule, and it can only move in one direction. KJ, what are you rambling on about? That's so confusing to me. Just imagine two one-way roads. I don't even care if you understand why this is happening. But DNA polymerase can only travel the correct direction. So one of them is pointed this way toward my fingers, so DNA polymerase can go this way. The other strand is pointed this way, so DNA polymerase has to go this way. There we go. It's hard to do it backwards like that. So DNA polymerase has to run in opposite directions, and that's a super big problem because the DNA replication fork, that's what this thing is called, is opening up. And so it allows access on what's called the leading strand, but not on the lagging strand. Now I have this fabulous video, and I encourage you to make a DNA narration where you narrate it yourself based on this particular video. I'm not going to take time to watch this video right now with you. I'm tempted to, but it's like 5 minutes long and it's just going to make my lecture a little bit longer. So I want you to watch it outside of class or pause mine, watch it, and then come back to what I'm talking about. So it says, watch this video on DNA replication before going any further. I'm going to keep moving on. Pay attention to the difference between leading and lagging strands. So let me get my image out of the way here and let's take a look. So here's a eukaryotic DNA, and we see an origin of replication, and it's moving in this direction. This one is called the leading strand. As fast as that DNA is separating, as fast as that, it's called the replication fork, as fast as that replication fork is opening and the two old strands of DNA are separating from each other, there's a new strand of DNA being made, and DNA polymerase is right there. As fast as it's opening, DNA polymerase is copying it. So that's called the leading strand, and you can see a bigger image of it right here. There's a primer that was added by primase, and now DNA polymerase is right here, and it's copying the one side. And as fast as helicase is unzipping the two strands, DNA polymerase is right behind it, adding the new DNA. So it's driving along this one-way road. The problem is the other side down here. This is a problem because if it can only travel one way and it's closed right here, how is it supposed to get on and copy the whole thing? So it copies it in little sections, and you just have to watch the video because it helps a lot. Looking at just a dry diagram like this, it makes it a little bit harder to understand. All right, so I'm sorry, I forgot there were a couple other things. The leading strand is made, we say this, it's made continuously, just one long strand in the 5' to 3' direction. DNA polymerase adds nucleotides as quickly as the DNA can be untwisted and unzipped. Now here's the problem. The lagging strand has to be made in short segments. So let me get my picture out of the way again because I want to. This image right here that I have in the corner, this is called a replication fork. And it's a good image for us to look at it. So the original leading strand, we'll pretend the leading strand is this strand right here on top. So the DNA polymerase is following along and it's copying it super fast. And as fast as the DNA is untwisting and unzipping, the DNA polymerase is copying it. But now the DNA polymerase has to move the opposite direction on the lagging strand because they're anti-parallel. So DNA polymerase, watch my cursor. DNA polymerase starts right here and it copies and copies and copies and copies and copies, and it gets to the end and it falls off. All done, right? Wrong, because in the meantime, helicase, while the DNA polymerase was driving to the right, helicase was unzipping the DNA to the left, and now a whole new section of DNA has opened up. The DNA polymerase running along here, it doesn't care, it's just copying and following the helicase all along. But the DNA polymerase that was going this direction, now it has to start over way back further where it has now opened up. Gosh, this is hard to explain. You really have to watch the video, everybody. Okay, so let's look at it on this diagram here. We've got primase, it added a primer. DNA polymerase comes in and it copies one strand and it gets to the end and it falls off. We're all done, right? Wrong. In the meantime, the DNA has opened up behind where it originally copied. So now a new primer is put down and DNA polymerase copies this new section that opened up. And when it reaches the section that it already copied, it falls off. But in the meantime, a new section of DNA has opened up. So a new primase attaches a new primer, and a new DNA polymerase has to copy that section behind the section that it copied behind the section. Okay, so forth and so on. So each of those little sections that it's copying are called Okazaki fragments, named after the scientist who kind of figured out what was happening in this process. So it says when the DNA polymerase reaches a primer, it stops. Eventually, the primers are removed and DNA nucleotides are filled in. So I think it shows it, I think kind of right here, they're showing it being removed and the correct DNA nucleotides are being attached. Now, this is a subtle, and I wish I could close up, and I want your eyeballs to be really close and look at this. This last enzyme right here, we haven't been introduced to this enzyme yet. This enzyme at the very end is called ligase, and I want you to look carefully. There's a tiny little gap right where my arrow is pointing. There's a tiny little gap in the blue new DNA. And the reason there's a gap is that that's the separation between the two different Okazaki fragments. They haven't been covalently bonded together because they were made separately. They were made at different times. So ligase's job is to come in and make a covalent bond so that it is all one long DNA piece and there's not little chunks of the DNA piece. So you got to kind of have a close up on that. It'll make sense when you see the video. I think that really explains it as well. All right, so again, this is a nice picture of the whole process, and the DNA ligase is labeled here. So this whole thing is called a replication fork, and that's this picture that I have in the corner of all these images as well. On the top, we see the leading strand, and the DNA is traveling in the same direction that the topoisomerase and the helicase are separating the DNA. So it's just cruising down the highway going as fast as possible. But the DNA polymerase on the lagging strand is going this direction. So it goes this direction and then it falls off the end and it's like, oh, I'm all done. Only in the process, topoisomerase and helicase have opened up highway behind it. So a new DNA polymerase hops on, it makes the Okazaki fragments. Eventually, the primers get replaced with DNA, pieces of DNA, so DNA nucleotides. And then this molecule, this enzyme called DNA ligase, joins the Okazaki fragments together. It makes these nice, strong, covalent bonds so that it's one continuous piece, even though it was just formed in chunks called Okazaki fragments. Whew, hope that made sense. All right, we're all the way at the end here, you guys. Termination, well, almost at the end. Termination is how replication ends. So the elongation continues until the replication bubbles bump into each other and all the DNA has been copied. The one problem, and you're already aware of it from some things that came up when we were when we were talking about cancer, the ends of linear eukaryotic chromosomes are difficult. And I'm not going to go into the details, but it has to do with the fact that there's a primer at the very end and then the DNA polymerase can't get on. So what ends up happening is the ends of the chromosomes get shorter by somewhere between 100 and 200 base pairs every single time the DNA is copied. So that's every time the cells divide. So they're getting a little bit shorter. So we have something called telomeres on the ends of our DNA, on the ends of our chromosomes that are made of repeating sections of DNA so that when the DNA gets a little bit shorter and a little bit shorter and a little bit shorter, that's unimportant DNA and it doesn't matter that it's not getting copied. Once it gets so short that we start getting into the nitrogen bases that matter, that are the genes that determine our traits, then the cell has to stop copying that DNA. And that's when we enter senescence. That's when our cells stop dividing and they say, okay, we can't divide anymore or the DNA is going to get damaged. And that's what leads to aging and old people diseases. All right, very last slide. Proofreading. So first of all, how fast students always want to know how fast does this process happen? It happens way faster in prokaryotes than it does in eukaryotes. There's many reasons. We'll talk about some of them. So in prokaryotes, it can happen as fast as 500 bases a second. So there's a DNA polymerase that's zipping through, replacing 500 bases or adding 500 bases in 1 second. In a eukaryote, it's more like 50 bases per second. So what's their error rate? How often do they make a mistake? And you can take a look at a mistake up here where there's two that bonded together and they're not bonded correctly. So the error rate is quite high, actually. It's about 1 in every 10,000 mismatches, which you're like, well, that's not very many. However, you have 3 billion letters in your DNA, so that means about 300,000 mutations every time a human cell divides. That's super problematic. So instead, or not instead, we have these proofreading enzymes whose job it is to identify mistakes and correct them. So you can see this enzyme here called nuclease. It's coming in, it's actually removing the nucleotides that were added incorrectly. Then the DNA polymerase, it's a different DNA polymerase, comes through and adds the correct ones. And then again, DNA ligase binds these sections together so that they're covalently bonded. And you would never know that there was a mistake made in the first place. The great news is that by the time these proofreaders come through, can you just pause for a second and think about how amazing it is that your body has proofreading enzymes? Like, that's wickedly crazy. Just be in awe of your body. Okay, so by the time the proofreaders are done, we're down to about 1 to 3 mutations per human cell division. So that's why, things like cancer increase with age. The more time the cell has gone through division, the more those mutations start to accrue, the more and more we add up. But only 1 to 3 per cell. That's pretty amazing when we have 3 billion letters in our DNA. All right, ladies and gentlemen, that's it. Let me know if you have any questions.